It's Barbara Melhart. I'm from the Department of Molecular Pharmacology, and this is a recorded class about immune responses against the novel coronavirus SARS-CoV-2 for the course Pathology and Immunology for first-year pharmacy students. Today's date is March 18th, 2020, and I would like to tell you a little bit more about how our immune system will handle such a new virus that we have no immunity against and we don't have any vaccinations to help us. So how does your immune system deal with it? So we know that this new virus mainly infects the lung and causes disease in the lung. So I wanted to start off with a little background on the structure of the lungs. So the lungs consist of airways and alveoli. The airways here transport the air and start off with the big trachea that will divide into smaller airways up until these smaller airways get to the alveoli, which is the parenchymal lung tissue where the actual gas exchange takes place. So our lungs breathe in a lot of possible dangers, including viruses, and we can protect ourselves against these viruses because the airways are coated with cells that won't allow entry of viruses. So these airways and alveoli are coated with epithelial cells. And depending on where you are in the lungs, these epithelial cells look a little bit different. So in the larger airways, it's mainly ciliated epithelial cells, as you can see here, that have cilia, that can help us with removing things that we breathe in by waving them up again towards the throat, so we can swallow them and digest them. To help with this waving mechanism, there are cells that produce mucus. These are goblet cells and mucus glands, and these produce mucus that trap viruses and prevent them from being able to infect our lungs. These type of cells are also present in the smaller airways, though the identity of the cells changes a little bit. There's some other cells coming up, and the epithelial cells are really different in the alveoli, because the alveoli is where the gas exchange takes place. So this is the air compartment, and here we have lots of blood vessels where the blood needs to be oxygenated, so the distance that the gases travel should be very small. So that's why this wall is very thin. And this thin wall is covered with type 1 epithelial cells that are really flat, and every once in a while you get a type 2 alveolar epithelial cell, which is a sort of a stem cell that generates new type 1 epithelial cells when these get damaged, and they also produce a little bit of mucus and coating of the alveoli. So these epithelial cells form a physical barrier to the infection. So they're very tightly connected, so a virus cannot pass into the tissue easily. They also make the mucus and peptide antibiotics that can kill microorganisms. And within these epithelial cells are also other immune cells that can help fighting an infection. So how does coronavirus then enter the lungs? Well, coronavirus, as shown here, has these spike proteins, and these spike proteins recognize a protein called angiotensin converting enzyme 2, which is expressed on certain types of epithelial cells. And this enzyme is normally involved in regulating blood pressure, but it's also expressed on certain types of epithelial cells where it transports amino acids into the cell. So in this case, the spike protein binds to ACE2 and transport the virus into the cell. So once the virus is in the cell, it will release its viral RNA and take over the machinery of the cell. So the enzymes that normally make RNA for the cell will start making viral RNA, and the parts of the cell that produce proteins start making viral proteins, and these are then assembled together into new copies of the virus. So the cell will create many new copies of the virus, and eventually the cell will explode and release these new particles, and then the infection can spread. So why does the coronavirus specifically infect lung epithelial cells? That's because the enzyme, or the protein that is used for entry into cells, ACE2, is expressed on cells in the lung. So here they looked at expression of ACE2 in different types of epithelial cells. So these are cells that are mostly present in nasal passages. These are mostly present in the lower airways, and these are present in the alveoli. And as you can see, alveolar type 2 cells have relatively high expression of ACE2, club cells in the lower airways, and in the nasal passages it's ciliated cells and goblet cells. So the coronavirus can infect these cells specifically very well, and that's why when you are infected you will have symptoms in your nose and in your airways. So what actually happens? So when a virus manages to enter epithelial cells, the incubation, it first starts with an incubation period, or sort of a stealth phase, in which the virus is in the cells and starts to replicate itself. The incubation time for coronavirus is not quite determined yet, but it's suggested that it takes 2 to 14 days for people to actually get symptoms. And these symptoms are caused by the fact that the innate immune system starts responding to the virus, and you get your first symptoms in the lungs. And then the adaptive immune system is also involved, and a whole inflammation response starts up. And in short, it's displayed here. So what happens is that dendritic cells in the lung will sense the virus and will go to the lymph nodes to get help from lymphocytes. The innate immune cells in the lung will start responding to the virus as a first line of defense. So neutrophils and monocytes will come in to help the local immune system and epithelial cells with clearing the virus. And then when the lymphocytes come in, they will start helping out these cells with clearing the virus. So there will be cytotoxic T lymphocytes that will kill any infected cells. There will be B cells that produce antibodies, and there will be CD4 positive T helper cells that will help other cells clearing the infection. And then if all goes well, the virus will be cleared between 7 to 28 days. So we'll go through this process a little bit more in detail. First, the stealth phase. The virus enters the epithelial cells, but of course our immune system has evolved and is alerted to possible danger. So our cells have many cellular alarm systems that can tell them that they are being invaded and they need to react. So an important class of cellular alarm systems are the toll-like receptors. These are there are many types, and they are either expressed on the cell membrane or within the cell. So since this virus goes into the cells, these extra cellular ones are not much use. But actually the ones that are in the cell will sense the presence of the virus. So particularly important in this case are TLR7 and TLR8, because they sense single-stranded RNA, and the coronavirus is a single-stranded RNA virus. So what happens when these toll-like receptors sense the presence of the virus? They will activate certain transcription factors, and in this case interferon regulatory factors, and that will lead to the production of type 1 interferons, so interferon alpha and beta, and these should lead to an antiviral state. Well, how does that work? Here we have the infected cell that will start producing type 1 interferons. This cell is already infected, so it's too late for this cell to survive, basically. But what it can do is tell its neighbors that it's infected and that they should prepare themselves to fight. So that's why it makes a type 1 interference, because all cells in our body have receptors for type 1 interference. So if this cell starts producing type 1 interference, this one uninfected cell will sense the presence of type 1 interference and know that it needs to start getting prepared for a virus. So if type 1 interferons then bind to the receptor, what happens is that a translation initiation factor that is needed for protein synthesis is phosphorylated, and thereby it's inhibited, so the virus cannot produce viral proteins. It will also increase the expression of RNases that can degrade viral RNA. So once the virus enters the cell, it cannot survive anymore. And it will inhibit viral gene expression and virion assembly. And altogether, this makes it a very inhospitable state for the virus. So the virus has little chance of surviving this, and it cannot replicate anymore. The importance of interferon signaling is shown here for the first SARS virus. So this is not the novel coronavirus, but the one from 2003. What they've shown in people who were infected with this disease, if they had a very early type 1 interferon response, that they could contain the viral infection and they had very mild disease. But in people that had a delayed response of the type 1 interferons, the viral titers were much higher and they had much more severe disease. Right, so that is the sensing and preparing the cells to defend themselves against the virus. But of course, you need more, and the cells that are being infected and destroyed will also generate cytokines and chemokines that will alert other immune cells. So the chemokines will attract neutrophils from the blood, and these neutrophils will phagocytose infected cells, and they will also phagocytose viruses, as you can see here. And they will go through a process called netosis, in which they will make sure that their DNA is being extruded out of the cell, so they can catch any loose viruses that are in the tissue. And this is possible because DNA is very sticky. So if they extrude their DNA, the viruses will get caught in this very sticky DNA. Monocytes can come into the tissue as well. These monocytes can differentiate into macrophages that can then differentiate into M1 macrophages that are very helpful in killing microorganisms. But they can also differentiate into macrophages that will help cleaning up the mess that is being made here, because especially these exploded neutrophils will cause a lot of tissue damage, and the M1 macrophages will cause tissue damage, so you'll get an inflammation here in the tissue that can be cleaned up by macrophages that develop into a different phenotype. But normally this should be where the virus stops, but in the case of coronavirus, this usually doesn't happen. Your immune system, your innate immune system that is trying to contain the infection here is not enough. So more help is needed, and that's when the adaptive immune system is being called for help. And the adaptive immune system are the lymphocytes, and those are present in lymph nodes. So this is a lymph node, and a lymph node has very defined zones. So these blue bubbles here are B cell zones. That's where the B cells are waiting around for a signal to get into action, and the T cells are stored here. So what happens is the lymphatic vessels that drain tissues will drain viral antigens, and they will drain dendritic cells that have eaten up viruses or cells that were infected with viruses. These all come into the lymph node and will alert the lymphocytes there. So first of all, what happens to the B cells? So the viral antigens will be transported to the lymph node through the lymphatic fluids, and the B cells can directly recognize these viral antigens. So in these blue areas, the B cells that are waiting here, some of these B cells will recognize these viral antigens and then become activated. As you can see here, here's a B cell that will recognize a viral antigen with its B cell receptor. It will then become activated and become a plasma cell, which will produce lots of extracellular antibodies. And what can these antibodies do? Well, first of all, they can neutralize a threat. So in the case of this virus, it can bind to the virus and prevent it from entering other cells. They can induce complement lysis. So if a bacteria has entered our body, it can be lysed by the complement, which is then activated by the antibodies. The antibodies can also help with opsonization. So viral particles that are coated with antibodies can be recognized by macrophages and then be phagocytosed by macrophages, and in that way be degraded. And infected cells that are expressing viral antigens can be recognized by the antibodies, and then the cell coated with antibodies will be recognized by natural killer cells that can then kill the infected cell. So that is what B lymphocytes do. But what do the T cells do, and how are they engaged? So here we have a dendritic cell that will go to the lymph nodes to get T cell help. And T cells do not recognize antigens by themselves like B cells do. They need antigen-presenting cells. So a dendritic cell is an antigen-presenting cell that will present the antigens to the T cell. And there were two types of T cells. We have the CD4 positive T helper cells. Basically, those are the lymphocytes that tell others what to do. They tell macrophages that they should kill whatever they've eaten up. They tell CD8 positive T cells, or the cytotoxic T lymphocytes, to kill a cell, and they tell B cells to make antibodies. And then we have the CD8 positive T lymphocytes, or the cytotoxic T lymphocytes. Those T cells can directly kill infected cells. So here's the dendritic cell with the antigen that it's presenting either to CD4 positive T cells or to CD8 positive T cells. If it presents to CD8 positive T cells, these will become activated and start dividing and go to the blood and then to the infected tissue, where they'll look for infected cells. And if they find an infected cell, they will kill it. The CD4 positive T cells, if they recognize the antigen presented by the dendritic cell, they will also multiply, go to the blood and go to the infected tissue. But there they will help B cells produce antibodies. They will help the CD8 positive T cells killing cells, but they will also help macrophages that have ingested microbes to kill those microbes. So how do these T cells recognize the presented antigen? And that was through a molecule called major histocompatibility complex, or MHC. Here's a CD4 positive T cell, here's a CD8 positive T cell. They both have a T cell receptor, but the CD4 positive T cells only recognize antigen presented in MHC class 2, and CD8 positive T cells only recognize antigens presenting in MHC class 1. MHC class 1 is present on all cells in the body. So all cells in the body can present antigens to T cells, and that will be a signal for the CD8 positive T cell to kill this cell, because the cell basically tells the CD8 positive T cells, I'm infected, I need to be killed, you need to contain this infection. Antigen presenting cells are the only ones that have MHC class 2 and can present antigens to CD4 positive T cells. And that's their way of telling the CD4 positive T cells that they need help with killing a microorganism. So a antigen presenting cell, like a macrophage or a dendritic cell, eats up viruses and then tells a CD4 positive T cell, we need your help because I need to kill this thing, what I've eaten, and the B cells need your help to make antibodies, and the CD8 positive T cells need your help to kill infected cells. So in this way, any cell in the body can tell that's infected, and antigen presenting cells that eat up lots of things can get extra help for a tissue when it's infected. So in short, here we have the B cell that can directly recognize an antigen and then activate and become a plasma cell which produces antibody. A CD8 positive T cell can directly recognize an infected cell and destroy it And a CD4 positive T cell is activated by an antigen presenting cell, and this antigen presenting cell basically asks the CD4 positive T cell to help it with destroying an infection, a virus, and to help the CD8 positive T cells with destroying infected cells, and to help the B cells with making antibodies. So how does a CD8 positive T cell actually kill a infected cell? Well, when the infected cell is presenting antigen in the context of MHC class 1, it can be recognized by the CD8 positive T cell. This will then get activated, and it will start exocytosing granules. So it will throw out granules from the cell, and these granules, as depicted here, consist of two proteins called perforins and granzymes. And perforins make holes in membranes, and granzymes induce apoptosis by activating caspases. So they're both in sort of particles together. These particles are taken up through endocytosis by the infected cell. And then once these particles are in the endosomes, the perforins make holes in the endosome, and then the granzymes can escape the endosome, go into the cytoplasm, and activate caspases, which will induce apoptosis. So what that looks like, you can see here. Here's a big infected cell, and these are two CD8 positive T cells that are sniffing around, looking to see if this cell is normal, and then we'll decide to kill it. You can see they're sort of sniffing and feeling their way, and then they will exocytose those granules, and that will lead to the cell being killed and completely dissolving. So this is an overview of what happens with the lymphocytes. Here we have the naive B cells that recognize the virus and become activated. Then they start to divide. You get clonal expansion. That's also when you see your lymph nodes grow, because the lymphocytes are activated. Then they'll differentiate into plasma cells that produce antibodies and will help with elimination of the virus. The T cells recognize the virus when it's presented by antigen presenting cells or dendritic cells. Then they will become activated, and they also go through many divisions, clonal expansion. Then they will differentiate into effector cells and help with the elimination of the virus. Once the virus is eliminated, all these extra lymphocytes go into apoptosis, but not all of them. There are a few of them that will turn into memory cells that will survive and will help out with a next infection. So if you're exposed to this coronavirus again, they will help with quickly clearing that next infection. And that is shown here. So this is the first time you see the coronavirus. The virus has basically a good chance of replicating because your immune system is not up to speed. But then your immune system manages to clear the infection, and the next time you're being infected, your immune cells, they know about this virus, so they can quickly respond. So you get a quick proliferation of your memory cells that will help with killing the virus, so your virus doesn't have as big of a chance to replicate. What do we know about immunity against coronaviruses? we know that the coronaviruses that cause the common cold in humans induce short-lasting immunity for whatever reason that is unknown, but up to a maximum of 3 years, and then your immunity against that coronavirus is sort of gone. So we don't know this for the novel coronavirus, what the immunity is like. That time will have to tell us. But of course, if the immunity is that short, because 3 years is not a very long time, it also means that vaccinations need to be repeated many times. But we don't have any vaccinations yet, so we'll have to wait for those first. So to get an idea of the disease features of the SARS-CoV-2 virus, or the disease called COVID-19, please go to the other class about this topic.